Everyone, this is Elad from Astrolabe Diagnostics, and today I would like to discuss analyzing Cytek Aurora data in Astrolabe. Specifically, I would like to talk about OMIP 069, which came out in late August 2020. This very cool paper came from a 3-person team at Cytek, and they describe how to do deep immunophenotyping of human PBMCs using a 40-color panel, which is quite phenomenal for flow cytometry. To me, the interesting figure is figure 1a, and this is the gating strategy for this panel. You will notice that it is much more complicated than what you would expect from traditional flow, just because you have so many markers. And this is a trend that we're seeing quite often now. As the panel increases, the gating strategy increases exponentially. This means that gating this data will be quite time-consuming and furthermore highly variable. For example, if we look just at this gate over here, if you move it by a few pixels, it will change the B cell gates by quite a bit. This is where Astrolabe comes into play. Astrolabe allows you to run your experiment as always, acquire your data, whether it's human or mouse, run it through your instrument of choice, in this case the Cytek Aurora, and then upload the FCS files to our platform, which takes care of all of the steps of the analysis pipeline, specifically cleaning the data, identifying different cell subsets, conducting unsupervised clustering, and, if necessary, identifying populations and biomarkers that change significantly across your experimental conditions. All of this is done automatically. There's no need to draw any gates, choose any your LNE algorithms, or set any parameters. Specifically, this is how the OMIP 69 experiment looks in Astrolabe. Just to give you some perspective, getting from the FCS files to what I'm going to show you here took about 15 minutes of waiting. So we have the 4 different FCS files, they're tagged with the donor IDs. The first thing I'd like to show you is the quality control tab. This shows us the percent debris and how many cells could not assign to any subsets here. Astrolabe identifies dead and debris cells for you automatically. And just to give you a brief look of how this works, when you run your experiment, you can download the reports, and within the reports, you can look at the different gates that Astrolabe uses. For example, here is how Astrolabe removes debris and doublets in this data. Then Astrolabe identifies the different cell subsets, and these are the default populations that Astrolabe found in this data. The process is conceptually similar to traditional gating. So in this case, Astrolabe using CD3 to identify T cells, then CD4, CD8, and TCR gamma delta to find these major types of T cells, and then within the CD8 and the CD4 using CD45RA, CCR7, and other markers for naive memory and Tregs. This entire mechanism is flexible, so it will adjust to your panel, and if you want to make changes, you can always reach out to us and we will work with you. Going back here, Astrolabe takes these subsets and organizes them in a dimensionality reduction view. This is similar to t-SNE or to the SPADE algorithm. And then you can click on any of these populations and scroll down to see their statistics across the different samples. So here are the subset frequencies, or to get a heat map that shows you the marker intensities, again, for each one of the samples, each one of the cell subsets. One last thing you can do, you can go into a specific sample view, and within that sample view to get a heat map of all the different cell subsets, all the different markers, and the expression of these markers across the subsets. Astrolabe also has extensive biostatistical capabilities. Unfortunately, there's only 4 samples here, so there isn't much to show in that regard, but I encourage you to reach out if you have any questions on that context. Thank you for your listening, and if you are planning to do any Cytek Aurora data, I encourage you to reach out and talk to us about your analysis plans. As I mentioned, it's a great instrument. It's a very exciting OMIP and panel, but be wary of the analysis, be wary of the gating, and the time investment in that regard. So I'm Elad at astrolabediagnostics.com. Please make sure to subscribe to our YouTube channel and to our LinkedIn page to get more of these videos.